Hello, anatomy and physiology students. I hope it's not too unpleasant to hear my voice again, and welcome to the first of however many classes we have left, our lecture notes. I do not have guided notes for y'all because most of y'all don't have access to printers. I would recommend that you take notes on this in your notebook. That way, if we have any exams or you plan to use this notebook later, say in college, that it will be helpful to you. Also, side note, you may hear cat noises in the background, and I apologize for them in advance. What we're going to talk about is the structure and function of neurons, which are the basic cells that make up the nervous system. It kind of helps to have an understanding of this before we go into the brain, which is going to be our next major topic. So, before we can talk about any of the function of a neuron, we need to look at its structure. As we've talked about throughout the year, both the anatomy, the structures of a particular organ or cell is very important to its function, what it does. So, we're going to look at different parts of a neuron and we're going to talk about what they do. Now, keep in mind that neurons are all about transferring information. Information goes in a neuron in one direction only. As you see down here, which is pointing out the direction the electrical impulse travels, and that electrical impulse is carrying information. So we're going to kind of go through this in the order in which this information goes through the cell for the most part. So information comes in through these structures called dendrites. They're these branching structures. They're actually named dendrite. Dendro means tree because they look like the branches of a tree. A neuron can have a few of these or it can have hundreds of these, and they can branch off to be able to connect to multiple other neurons. This is what receives information. So it gets information from another neuron or from an organ, from a sensory receptor, and it's going to take in that information. That information is going to be processed in what's called the soma or cell body. All of the information coming in from each of the dendrites is going to get added together in the soma. And if it reaches the right conditions, it's going to travel to the structure called the axon hillock, which is basically kind of a bulge on the end of the cell where this next structure, the axon, is going to come off. This part of the cell is really important because it's what decides, is the cell going to send off a message or is it going to stay silent? If it does decide to send off a message, it's going to send off a message along a usually fairly long projection called the axon. Now, depending on what the next neuron or organ or cell in the chain is, this axon can be either very short, few millimeters, or it can be very long. For example, if you have a 6 foot tall person, their longest axon extends from the base of their spine down to their foot. If you're looking at an organism like a giraffe, that's a very long axon that's going to go from the base of their spine down to the bottom of their feet. And so these axons are different lengths depending on where they are in the body. Now, in the peripheral nervous system, axons tend to be longer because they have greater distances to travel. In the central nervous system, you can have some long axons and you can have some short axons depending on the function of the cell. And then just because it's such an identifiable structure in any cell that you're looking at, the nucleus is there in the center. When I say it does nucleus stuff, that's because it's doing stuff involved with DNA and controlling the cell, and that's more of a topic for when you're learning more about DNA and such. We're going to focus on larger things. So, what you may have noticed is these kind of bulges surrounding part of the axon, and this is basically what's called the myelin sheath. A sheath is a protective covering. You can think of it almost like a sleeve. So if any of you guys are interested in fashion, there's a style of dress called a sheath dress. It tends to be fairly form-fitting. You can sheathe your sword, meaning put your sword away in its scabbard. So it's a protective covering. What its job is it increases the speed of signal transmission. A signal in a neuron is called an action potential. And when you have myelination, that is going to significantly increase the speed of transmission, which is really important if you're trying to have split-second decisions made. If you are trying to transmit signals across a very large distance, like say from a giraffe's spine down to its feet. And so this is kind of better than the other possible way this can happen. You can either have um, an axon get wider, or you can have it be sheathed in myelin. And if your axons got too wide, then your nerves would be much bigger. This also allows you to save space. And these myelin sheaths, which are basically layers of fatty membrane, are serving as an insulator. So all of this stuff that you see here, that's basically wrapped up layers of membrane. So it's like the cells that make this, which are oligodendrocytes in the central nervous system and Schwann cells in the peripheral nervous system, they basically just wrap themselves around the axon and make themselves a protective cover. Um, Basically, they speed up the transmission of signals instead of the signal having to pass from here to here to here to here to here. The signal jumps from one gap in the myelin sheath to the next. And this jumping kind of conduction is called saltatory conduction. So if you run across that when you're in a college anatomy class or when you're in like a college biology class, that's what it's talking about. This jumping conduction from point to point to point. So it's going to speed up transmission that way. So let's talk about these signals that neurons send. So neurons send messages electrically. You can carry electrical energy in several different ways. In your devices, like whatever you're watching this presentation on, those electrical signals are carried by electrons. On the other hand, in your nervous system, you don't have a bunch of electrons flowing around. Instead, you have these charged particles, these ions, usually sodium and potassium, although calcium and chloride can have important roles as well. So when these charges move, that carries electrical energy and that causes an electrical current, basically. So, we're going to look at this just very basically. We're not going to go into a lot of the details just because it's a lot of nitty gritty information that I'm not sure you need to know for now for anatomy class. So, if you have an axon, what you're going to have inside of it is a whole lot of potassium and very little outside. Inside the cell, you have a little bit of sodium. Outside the cell, you have a lot of sodium. Now, as long as there are no gaps or holes in the cell membrane, which is represented by these thick lines right here, that's membrane, then, these concentrations are going to stay constant. But if you could open up a doorway, and that would allow molecules or ions to move through freely, what you would find is that sodium would move into the cell and potassium would move out of the cell. They would just diffuse naturally, go from where there's more of them to where there's less of them. And that's very important because what the cell can do, and there's a lot of negative ions inside. The cell can facilitate or aid the movement of those charges. So before we get into it, there are a couple of terms to look at. We're going to talk about the potential of a cell, and that's basically its voltage or its charge. And I'll try and use charge wherever possible, just to help you understand kind of what's going on. A depolarized cell is becoming more positively charged. And then to return back to a negative charge, a cell becomes repolarized. So, this membrane charge is going to be created by concentrations of ions, and it's going to be changed by moving those ions around. And there are a couple of ways those are moved around. There are these channels that will let stuff leak through. They're pretty specific, so they're like a dog door or a cat door. For example, in my house, I have a cat door. It is small. It will not allow my parents' dog to get in and out of my house because she's a 50-pound shepherd mix. But it will allow my smaller cats, even the fat one who's like 20 pounds, to get in and out of the house. It is specific, it is a cat door. On the other hand, if I got a larger door, that might be more specifically a dog door. And so, just like that, these channels allow specific ions to enter and to leave. And so they allow charges to move across the membrane and change it to become either more positive or more negative. And then you also have a pump, the sodium potassium pump, which will actively pump material into or out of the cell using energy. What's going in and out is not important for our purposes right now. What's important to know is that at a certain charge, you generate something called an action potential. An action potential is a very rapid change in the charge of the membrane. We call them spikes as well. And what happens is a stimulus comes along, it's going to make these ion channels open, and it's going to cause a local change in the charge of the membrane. Then your sodium potassium pump resets everything so that way you can have another action potential come along. So if we're measuring the changes in charge of a cell membrane as you go through an action potential, this is what it looks like. So for some reason, my entire screen is not showing up, but the x-axis is in milliseconds. And then the y-axis is in millivolts. And you can see that this down here, what's called the resting potential, is the normal charge of a cell. It's kind of negative. You don't need to know the exact number. What's important is just to know where it is. Now, when a signal comes in, it will change the charge of the cell, make it more positive. Once it reaches a certain point called the threshold, it will cause an action potential to happen, which is a rapid depolarization or becoming very positive, and then a rapid repolarization going back down to negative. There's also a short, what's called hyperpolarization. Notice that its charge dips down below what it normally is. It gets a little extra negative, and then it goes back to normal. Only once you've gone back to normal or back to resting potential can you actually have another action potential come through. And if possible, I'm going to look for and see if I can find any good simulations to let you guys see this happening, but don't pay attention to the molecular details when I do. So, action potentials are started at the axon hillock, which makes a decision. Am I going to fire an action potential or am I going to stay silent? And then that action potential gets passed along in one direction only, and it moves from the cell body down to the ends of the neuron. And once they reach the end of the neuron, we're going to have information pass from one neuron to another at a location called a synapse. So, a synapse is an area where one cell meets another. The axon ends in something called a synaptic terminal. Here it's called an axon terminal. And what's important is the electric signal that's being passed along by action potential now changes to a chemical signal that's being carried by these molecules called neurotransmitters. And if you hear about chemicals in the brain, these are likely the ones that you're hearing about. And we're going to talk about them more on the next slide. So these neurotransmitters are these small molecules. They get released by the cell that's sending a message. And when they get released, they're going to interact with these receptors, which are just there to receive messages on the next cell. And so that's going to be from axon to dendrite. There is a small physical gap in between these two neurons, and the molecules will have to travel across that in order to reach the dendrite of the next neuron. But when they do, they can either trigger that neuron to fire or they can prevent it from firing. It just depends on the neurotransmitters that are in use, as well as the kind of other inputs that a cell is getting. So, there are a number of different neurotransmitters. There are many more than exist up here, but these are kind of the big ones. And while this is not an entirely accurate diagram, it gives you the general gist of what you look at when you see these different neurotransmitters, the general responses that they're involved in. But this does not by any means mean that, oh, this is the only neurotransmitter involved in this and this is the only function that this neurotransmitter has inside the body. So there are a number of excitatory uh, neurotransmitters. Acetylcholine is something we've already talked about. This is at the neuromuscular junction. So acetylcholine is essential for your muscle cells to fire. It's also involved some in learning and memory and say the brain. Dopamine is involved in a number of things like reward or pleasure. So a lot of the like narcotic drugs that have addictive properties will activate the dopamine system or mess with dopamine levels in the brain, which is why there's such a reward sensation of taking them or a good feeling from taking them because it's activating those pleasure centers in the brain. Serotonin is here, it's called the mood neurotransmitter, and that's because several mood disorders, including major depressive disorder, are linked to serotonin and dysfunction in its release, either having too much or too little, usually. And so a lot of common antidepressants will target serotonin in the brain. Epinephrine and norepinephrine are other names for adrenaline and noradrenaline. It just depends on whether you're British or whether you're American, which one you call it. Adrenaline, the fight or flight neurotransmitter, that's what's released by your adrenal glands, and noradrenaline is implicated in concentration. Then an inhibitory neurotransmitter is GABA. They call it here the calming neurotransmitter, but what you find in somebody who has, say, a seizure, which is a very, very heavy storm of electrical activity inside the brain, their GABA neurotransmitter is often not functioning appropriately to kind of calm the activity down. There is such a thing as too much activity in the brain, and it's called a seizure. Glutamate is also involved in it's involved in memory. It's also a very common neurotransmitter throughout the nervous system. And endorphins are involved with feelings of great pleasure, happiness, etc. That's why they're called the euphoria neurotransmitter here. Like I said, this isn't a complete list. If you ever decide to go into neuroscience, there are a ton more neurotransmitters to learn about, and they have all these different activities, which are really interesting. So we've talked about all these neurons, but now we're going to talk about the cell that makes up the cell type that makes up the majority of the nervous system by both volume and number of cells. Glial cells are sometimes like, ignored by people. They're like, oh, these are the 90% of your brain that you don't use. But that's not true. These glial cells are very important. I think of them as personal assistants for neurons. Neurons are so occupied with the task of taking in and processing information, they don't do a lot of stuff to take care of themselves. So these glia cells are there to help them out. It's like you have a CEO of a corporation. Yeah, there's a single CEO who's doing work, but behind them you have, 10 to 100 people who are helping to keep things running around them so they can concentrate on running the company. Similarly, you have a principal in a school who has a large staff. They have assistant principals, they have office personnel, etc., who kind of help them run things. So that's what the glial cells are, personal assistants. In the central nervous system, they have specific tasks. For example, the oligodendrocytes shown here help to myelinate the axons, so they help speed up signal transmission. They act as an immune system, which is what these microglia do. Your brain has an extra barrier that kind of helps prevent things from getting to it, since your brain is such an important organ. So your immune system can't even actually get into the brain. So it has its own separate immune system, which is made of these microglia. Your brain, while it has blood circulating through it, also has another fluid outside of the cells called cerebrospinal fluid, or CSF. We're not really going to talk about it, but it's made by these guys, ependymal cells. And then astrocytes are kind of like those direct personal assistants. They are going to help regulate the activity of neurons. They help clean up extra chemicals that are floating around. They can help maintain the right concentrations of ions, and so on and so on. In the peripheral nervous system, you have a different type of cell that's going to myelinate the axons, and that's these Schwann cells right here. And then each ganglion that's in the peripheral nervous system is surrounded by these satellite cells. There's still a little bit of debate about what they do exactly for the satellite cells, but I mean for the ganglia, but they tend to cover the ganglia completely, and it's thought that they might have a function a lot like an astrocyte, kind of doing a lot of functions to help keep the environment of the neuron uh, constant and in the right, conditions so that it can very easily and effectively transmit information. So that is the end of our lesson for today. Next time, you will get a lesson on the brain.